Hello. This video we're going to be talking about chapter 4. We're going to go into the basics of RNA and DNA. Like that. So DNA, I'm sure you guys know this by now because it's deoxyribonucleic acid, while RNA is simply ribonucleic acid. So the difference is actually going to be this OH group versus H in the sugar portion of the DNA, stuff like that. So the numbering, which will become important here, stuff like that. So for carbohydrates, you typically you're going to have to start at the end of the chain, but typically you're going to start at the anomeric part. That's where the aldehyde is for aldo sugars. With that, so the numbering is going to go 1, 2, 3, 4, 5, so like that. So when we talk about 3' and 5' positions later on, it's just the numbering for this. Now, why is there a prime? Well, because there's another numbering set for the bases, which we'll talk about in a bit. But that has their own set of numbers, and so to let you know, we're talking about the base versus talking about the sugar, they put the prime in there, with that. So this is regular ribose, and this is the deoxy is literally just the only difference is missing that oxygen there. Okay, so DNA is going to be encoding for information, RNA or for long-term storage, I should say, and for RNA it's going to be more short-term. So with that, being able to do this. And so why does DNA use the deoxy form where RNA uses the regular form? Well, one, it's easy for the proteins to interact, for this, the components of the cell to be able to tell the difference between RNA and DNA, stuff like that, because they can detect the presence of that OH group. But also the deoxyribose is also less likely to undergo the self-cleavage. So if you look at this, so this OH can come in and attack, do a nucleophilic attack here to the phosphate. If that collapses back down, you can actually clip that bond there, stuff like that. So not a big deal for RNA because we can always just make more, but for DNA we need to be able to avoid that because that would just happen just in aqueous solutions. So for long-term storage it's easier to use the DNA. Okay, now the nucleotide, so the monomer units, so the links in the chain, okay, for the DNA molecules are going to be have 3 components. You're going to have the base, which is the A's, the C's, the G's, and the T's, which we'll talk about in a minute. We have in this case, the deoxyribose. For RNA, you'd have the ribose molecule, and then you also have the phosphate. Okay, so for that, you're going to need those 3 components to make the RNA, and they go, sort of sugar, phosphate, sugar, phosphate, sugar, phosphate, with the bases extending away like this. Okay. Now, the bases themselves, okay, they're in 2 different classes, the purines and pyrimidines, stuff like that. So this is a general purine structure, this is a general pyrimidine structure. The way that I sort of remember it is little name, big structure, big name, little structure. So both RNA and DNA use adenine, guanine, and cytosine, but the RNA uses uracil, while DNA uses thymine, and it's a way for the cell to be able to recognize RNA versus DNA, like that. So it's a nice way of doing it. Okay, so you will see here the difference between sometimes here nucleoside versus nucleotide. The only difference is the presence of a phosphate. If it's a nucleoside, no phosphate on the molecule. If it's a nucleotide, you're going to have a phosphate usually at the 5' position, hanging off the 5' OH. So something like ATP would be like this, stuff like that. But you can also technically have it down here, or you can have it some other place, stuff like that. So the only difference between nucleotide is a phosphate molecule hanging off. Okay. So the way we traditionally read these is from what's called the 5' end to the 3' end. And we just rattle off the bases because the rest of this is going to be common. So really the only difference between the links in the chain is this base here. Now the reason why this is called the 5' end is because the 5' OH here doesn't have anything attached to it. So remember the numbering goes 1, 2, 3, 4, 5. So this OH would be just at the 5' end would just be either have just a phosphate with nothing else hanging off it, or just an OH. And then it goes 3' to 5', and then 3' this 3' OH to this 5' OH, and you go on, and the 3' end it's going to have either a phosphate or just an OH here, and that's going to be free, and so that's what we call 5' to 3', and we just rattle off the bases. Okay, so in this case it would be A, C, and G, stuff like that. So on and so forth. Okay. And so, but one of the things that happens is, we talk about the bases, we talk about the sugars, but we rarely talk about the actual phosphate in the backbone. And there's a reason why there's a phosphate. One, the negative charge does a couple of things. One, it helps to maintain that water solubility, right? Charged things are going to be much more soluble in water than uncharged things. But also this repulsion that's happening by putting these negative charges right next to each other, it actually forces the molecule to be out, right? Because they want to, it's just like having a bunch of north magnets trying to put together, right? They're going to repulse. And so by having this, it stretches out the molecule, okay? Stuff like that. So, and people have actually gone in and made these neutral, okay? So they're not, so there's no charge to them. And what happens is it just collapses. It just does this great hydrogen bonding thing, and it all just kind of collapses on itself. But by putting the phosphates there, okay, you can help stretch it out. So, okay. Now, the structure of DNA, okay, so it's two anti-parallel strands of nucleotides, and they're going to be complementary to each other. And the way that those things work is sort of, there's what's called the double helix, and it's sort of like two ribbons that are wrapped around. It's not a spiral, but it's more like a staircase going down. Okay, so Watson and Crick are credited for discovering this, but they actually used Rosalind Franklin's data to be able to do that. So this is the X-ray crystal diffraction pattern that they would use to do this. So you would shine X-rays on there, and and when it hit an atom, it would fragment out. It would bounce off, something like that. And you can do the calculations to be able to do this. And they looked at this picture, and realized, oh, it must be a helical structure on this. So they didn't. There's a popular myth that they stole it. They did not steal it. Rosalind Franklin gave it to the head of the institute because she was leaving for another position, and then he showed it to Watson and Crick, and they immediately realized what it was. It is pretty sad that she never got the Nobel, because there was certainly space for her, but that's a whole nother issue. So, but it does have this double helical structure with this with these complementary things. So you can sort of see here, if you sort of zoom in, sort of a ribbon structure. If you do space filling, you can see there's two grooves. There's a major groove right here, so it goes there, and then there's a minor groove right here, and then right there, and stuff like that. And so these are going to be important for other processes being able to look at. So for recognitions for proteins and whatnot, stuff like that. So now the complementary bases. Okay, so you're going to have guanine goes with cytosine, adenine goes with thymine, and it's all about hydrogen bonding. This is the sort of classic Watson and Crick pairing. Okay, they knew something was probably up, and this is that because if you look at the A to T ratio, certainly in human beings it's 1-to-1, right? So for every one of these, right, you're going to have one of those because you need them to be complementary. It's not quite, it's not completely 1-to-1 most of the time, but usually it is. Now, the way that I remember it is if you put them in alphabetical order, the outside's complementary, the insides are complementary, stuff like that. So this is a little bit stronger bond. So GC, because it's got 3 hydrogen bonds. AT is a little bit less, a little bit weaker. So if you have a lot of G's and C's, it's going to be in your structure of DNA. You're going to have it's going to be kind of much more difficult to pull it apart, something like that. So just something. That. Now the way that these stack is these things are aromatic. Okay, so when things are aromatic, they're flat, and so they're going to stack like this. So you're going to have these two bases, the hydrogen bonding with each other, and they're going to be completely. That whole thing is going to be flat. And so what's going to happen is they're going to stack right on top of each other. Okay, and so you've got a couple of different things. You got van der Waals interactions. Okay, and you've got the pi interactions here. So the pi orbitals here in this aromatic group are going to want to interact with the pi orbitals down here, and so on and so forth. So again, it's going to be stacking like plates in a cupboard like that. Okay, there are a couple of different forms. It's almost exclusively going to be the B form. This is when it's hydrated, so this is pretty much what you sort of assume it's going to be. There also, if you have a dehydrated form, it does do, changes slightly. So with that, so the B form is the normal sort of Watson and Crick form. It's got the major grooves, minor grooves, things like that. The dehydrated form, if you notice, it's a little bit more open, sort of puffed out a little bit. So like that. So, but again, the way that it coils is similar to a right-handed screw, like literally as a screw, like nuts and bolts type screw. So like that. So there is a Z form that actually does a left-hand screw. They don't know that much about it. They know that it exists, and it has this weird left-handed screw, and it's kind of crazy. Stuff like that. So I wouldn't worry too much about it. What you're going to have to know is mostly this B form. Okay. Now, how does it hold the information? In eukaryotes, it's going to be in a chromosome with it. So in eukaryotes, right? So inside the nucleus, you're going to have these sort of chromosomes. The prokaryotes, right? So they're going to have, in this case, they're going to have circular DNA, and it can come in two forms, right? So it can either come in this sort of more relaxed form, regular circular, or you can get these what are supercoiled. So the way you can sort of imagine is these are the headphones. You know, if you've got corded headphones, this is what it's like when they're in your ears, and this is what it's like when they're in your pocket, right? They're all sort of coiled up. But you need to have that so we can get all the DNA packed into such a tiny space. Okay, so you can have. So we talk about DNA being double-stranded, and that's usually the form it is. But RNA, as it turns out, is mostly single-stranded, stuff like that. So, and you can have single-stranded DNA. Okay, and it can be self-complementary. Okay, so if you have a single-stranded DNA molecule, if you notice here, you've got some complementary parts. Now this is, then there'll be a loop and come back. Now this is, don't assume that this is flat. Okay, stuff like that. So you're still going to have this is coiled, but it's just a flat representation of it. You can get, just a simple stem loop like this, or you can get some very complicated looping structures, right? So where you have, these are complementary, but you have a little pucker, where it puffs out a little bit. In here where there's not complementary, here, these sorts of things, and these things can pack very tightly. And again, it's not flat, but this is a nice way to represent it, to see how everything is complementary to each other. Okay. So the double helix, this was fairly controversial, but trying to figure out how DNA is passed on from one generation to another, and it turns out it's semi-conservative. Okay, there was other things where there's conservative, there's semi-conservative. There's also where the DNA had to be completely destroyed and then remade each time. But it turns out that they probably take one of the simpler approaches and do the semi-conservative. And all that means is that the original parent DNA molecule is still going to get incorporated. It's just going to get replicated. So after the first replication, right, one of these strands is going to go on this side, one of the strands is going to go on this side, but the strand itself doesn't get destroyed. Okay, when these replicate again, these original blue ones are still going to be around. Okay, but now these magenta ones are going to be making new ones, and so you don't lose this DNA. Okay, it's still there even after the replication. It's going to break apart, sort of come apart, and they're going to fill in the new DNA strand. So okay, so DNA melting, stuff like that. So that means all that means is the DNA coming apart. The easiest way to do that is to actually just heat the DNA up. So at some point, this is what's called the melting temperature, where sort of half of it is well, I was going to say, so the relative absorbance, right? So you're going to absorb where it's double-stranded versus single-stranded. The absorption at 260 is going to be a little bit different at this point here. That's considered the melting point, where it's about that the absorbance is halfway, and all that really happens is the DNA base is there's enough energy in there so that they pull apart to go from double-stranded to single-stranded. That's all it is. As soon as you cool this down, they're going to come right back together, and they're going to form complementary bases, things like that. So, and it's just going to reform. So when you cool it down, that's called annealing, where the strands come back together. Okay. So how do we make new strands of DNA? Okay, we're going to use one of the strands. Okay, and we're going to use what's called a DNA polymerase, and that basically adds the individual components one at a time to build up the chain. Okay, so with that, so the first thing it's going to use are these the deoxy ATPs, so the nucleotides, triphosphate nucleotides. Okay, so with that, so these are the individual building blocks. Now it's going to know what to put on next, because it's going to say, okay, well here's a here's a T, so I need to grab a DATP to put in there. Now for the next strand, right, next up, we're going to have a C, so I better grab the G here to put it in there. And so it uses this this strand to be able to know what to make on this strand, and the way that it's built up is 5' to 3'. Okay, so what does it need? It needs the triphosphate monomers, as well as magnesium ions. So this is the magnesium cofactor. Okay, so when this leaves, after you add this, you get pyrophosphate. So with that, so two phosphate molecules put together, this can go on and then get broken apart into individual phosphates, and that's actually very good with thermodynamics. We'll talk about thermodynamics in a bit in a future chapter. So for that, but it helps to make sure that the reaction is energetically spontaneous, or exergonic, if you want to say it correctly. Okay, so this strand here is going to tell you what gets next. Okay, so you need to have the complementary base pair put in next. Okay, now the thing is, the DNA can't start from scratch. It has to have a short stretch of either DNA or RNA. It has to have what's called a primer, stuff like that. So it can be a few DNA bases. I mean, technically it could be one, but you're going to need a couple of base pairs in here to be able to start it off, and then it can start adding on to it, but it can't start from scratch. Okay, now there are RNA viruses. Okay, so with that, so these are the classic ones are the flu virus as well as the AIDS virus, and so these things are packed with RNA, and what those do is, when they get in there, the DNA gets squirted into the cell, and that along with a protein called reverse transcriptase, and that actually converts RNA to DNA, stuff like that. So and then that DNA then goes on to be able to use DNA polymerase to be able to make the new copy. It can hijack the cells, DNA polymerase and all that sort of stuff to make copies of itself and stuff like that. So, but this is just something for these retroviruses, and so this is why this is a nice target for those types of viruses, because it's only seeing viruses, so you can. It's not like you have a reverse transcriptase in your genome, so it's not going to mess up your the normal functions in your cell. So okay, so transcription. Okay, so this is the process of taking DNA to RNA. So the reason why it's called reverse transcriptase for the RNA is because it's going from RNA to DNA, so it's going in the reverse. Now there's a really nice set of YouTube videos, and I would highly recommend it. It's through the DNA Learning Center, stuff like that. So I would highly recommend doing this, because it's animation. It's using the actual structures of the components, and it talks you right through it. And it's a wonderful visual for being able to sort of see all the components and how this is going on, stuff like that. So, but the first step of this is transcription. Okay. So I would highly recommend this. They've got a whole bunch of different videos. There's one on transcription. There's one on translation, DNA replication, the whole kit and caboodle. So, okay, so there are actually three main types of of RNA. There's the messenger RNA. That's the one that actually transfer the information from DNA to RNA, so that it can then go into proteins. Oh, I forgot to mention, this is the, what is it? I can't remember if it's Watson or Crick called this the central dogma of life. Right? You're going to take the DNA, make this into RNA, and then that RNA can be used in the proteins. That first step is the transcription. Okay, so the messenger RNA is the one that's copied directly from the DNA strand. Okay, it's going to be single-stranded. Okay, and that's what we're going to use to say what protein is going to be made. Okay, the transfer RNAs or tRNAs. Okay, those carry the amino acids that we use to make the protein. So those are involved in translation. The ribosomal RNA. Okay, so these are the major components of the of making the protein. Okay, so the first thing that happens, right? So the RNA polymerase. This is going to be very similar to the DNA polymerase. Okay, you're going to need the nucleotide triphosphates. Now these are the regular ones. These are not the deoxy ones. So it's regular ATP, CTP, GTP, and TTP. Okay, again, it's going to use that single-stranded DNA template, and you can use magnesium, although manganese can also work as well. Now the nice thing about this is you don't have to have a primer. The RNA polymerase can start from scratch. It can just go in and start making RNA. Okay, so how the heck does it know? Well, there's promoter regions just upstream of where you're going to want to start making your DNA. Okay, so it knows to grab at this spot, and then here is where the DNA is going, or the RNA is going to start making it. So there's a place where the RNA can clamp down and then come in and start making your RNA. Okay, so there's different promoter regions to know. Okay, so there's promoter regions. There's also terminator regions. Okay, so you're going to have at the end of the coding region, you're going to have this what's called a poly A tail. It's literally just a huge string of A's, and it's going to knows when it hits that it's going to fall off. Okay, and then voila, you've got yourself your full-length RNA. Okay. Now the next thing, all right, so the next part is called translation. So this is where we're going to take RNA to protein. Now, if you can't remember transcription or translation, one, you can go alphabetically. Transcription comes before translation alphabetically. The other thing is, I always remember about reverse transcriptase, right? That's going from RNA to DNA, so that must be transcription. The other one is for translation. We always hear about what's called, in proteins, you always hear about what are called post-translational modifications to proteins, stuff like that. So that means after you've made the protein, if you put things on there and get it to fold correctly. So I always use that term to remember, oh yeah, translation is the one from RNA to protein. There's a whole bunch of other ways you can do that, but those are a couple. Okay, so for this one, okay, we're going to be reading this. It's technically the RNA code, which comes from the DNA code. Okay, to make to make the proteins. Okay, so by that, the way you do that is this 3 base pair codon, like that. And so if it encounters, say, a UUU, it knows to put in the phenylalanine. If it's a UCU, it knows to put in a serine. If it encounters the CAU, it does the histidine. And these are all done 3 base pairs at a time. Okay. So with that, so it knows. So how does it know which one? So how does it know that UUU should be phenylalanine? Well, it uses, this is the transfer RNA. Okay, the amino acid is this at this end, and then down here at this other part of the DNA, or the RNA is going to be what's called an anticodon. And so if this is UUU, right, this is going to be AAA, because that's going to be complementary to the U's. Okay, it's just like T's, right? So with that, so, and if you had, say, CCC, right, this would be GGG, and it would encode for a proline. Okay, so with that, so, and if you had, say, AAA on here, right, it would, the anticodon, right, would be UUU, things like that. So that's how it knows. It knows put it 3 at a time. It examines it 3 at a time and puts these things together, and it starts building up the chain 1 at a time. Okay, stuff like that. So this amino acid ends up getting added to the end of the chain, and then another codon, the tRNA comes in, reads the codon. Oh, I need this one, and puts that on the chain. You literally just start making the protein. Okay, so the thing is, so 3 bases equals 1 amino acid. Okay, there's no overlap in the codons. It goes 3, 3, 3, 3, 3, 3, 3. Okay, there's also no gaps. Okay, so it doesn't go 3 and then like an A, and then 3 and then and something else, and then 3 and something else. No. It's just 3, 3, 3, 3, 3. Okay, you're going to read that sequence from the 5' end of the RNA or DNA. It reads it from the 5' to the 3'. Okay, so like that. But there are some degenerate, right? We have 20 amino acids, but there's 64 possible codons, right? So like that, and so we get what's called the wobble base. Okay, so if you look here, that's the 3rd base. Okay, so it really only matters that you have a C and a U. This one doesn't really matter. Okay, it doesn't matter what this is. If you have a C and a U, it's gonna be leucine. Okay, if there's a CC here, it doesn't matter what this is. It's going to be a proline. Okay, now it does matter if it's if you have CA. Well, then it doesn't matter if it's a U or a C. That's going to be histidine, but it does matter if it's an A or G because that's going to be glutamine. Okay, so you see this a lot where it's the 3rd base pair that changes a lot, but they're going to be clumped together. Now the reason for that, there's origin of life stuff to say like they were originally 2, just 2 base pairs used as a codon, and then eventually evolved into 3. I think that sort of makes sense, but take it for what you will. Okay, so how does it know where to start? Okay, well, 2 things. There is a start codon. Okay, it's going to be methionine. I think we can get into that next. Yeah, there's start and stop codons. Okay, so in this one, it's in both cases it's going to be methionine, which goes for the DNA it's ATG, be AUG in the RNA. Okay, so like that, and so so it knows if it sees that, that's where we're going to start. Okay, some use some other ones, but 90% of the time it's going to be an ATG. Okay, so you're going to have a place where it's going to know how to bind, and then it's going to start reading this, and then the first ATG it sees, it's going to start putting there, and then start going, adding the forward. Okay, so the AUG here, or ATG from the DNA, that's going to be the methionine. Okay, and then you've got 3 stop codons, right? So when it hits this, it's going to know, okay, I'm going to stop, not going through there. Then the DNA parlance, it's TAA, TAG, or TGA, okay, or the RNA versions right here. Okay, now one of the things that happens with this is that in eukaryotes, okay, you have what are called introns and exons. Okay, prokaryotes, you don't have this. It's just gonna, when it makes the RNA, it's just gonna make the whole thing. It's just gonna make the gene, and that's it. But for prokaryotes, you're gonna have these introns, which I always remember because they're inserts. That's how I remember it. Okay, it's just this blue part that encodes for the actual protein, but it's got these sort of intron inserts in there for just extra DNA. Okay, and so what happens is during the process, in the first steps of translation, this is actually going to get fully processed to remove these inserts, so you can actually get the regular mRNA. So what happens is it sort of loops these things out, snips that out, puts this together, snips that, sort of pinches those off, and so here you have the mRNA. Now for things like yeast or whatever it is, it's not going to have a whole lot of introns, but if you're trying to, say, clone out a human gene from the DNA, it's going to have a lot of introns in there. So what you end up having to do if you want to clone out a gene, you actually have to isolate the mRNA, and then, because it's already been processed, the insert's out, and then you can use it. Okay, so why introns? Well, there's a bunch of reasons, but we're just going to go over 2 stuff like that. So one is called exon shuffling. Okay, so what it can do is it can shuffle this portion. It can take this portion from this protein and and this portion from this protein, this portion, and put it together, and you can, these inserts allow it to sort of shuffle these things around, so then you can make new proteins. So it's a way of making diversity. It's a way of making mutations, for Darwinian evolution. Okay. The other thing is what's called alternative splicing. Okay, and so this one can be seen in B cells, right, that make antibodies, stuff like that. So when it's dormant, okay, it's going to be making this version of the protein, okay, and it leaves in this part of the protein, some of that of the antibody protein that's an anchor inside of the cell membrane, okay, so with that, and so during while it's dormant, this is the full length protein, okay, with this part in there. As soon as it activates, what it does is it says, oh, now we're going to make that into, we're going to chop that out as an exon. So now we're going to make the protein like this. It doesn't chop this off. When you're going through translation, now it makes this form of the protein, stuff like that. And it's this part here that would have anchored here is actually got knocked out as an exon. So, or excuse me, as an intron, it's gotten lopped off. So this way, you don't have to have two versions of the antibody encoded in your DNA. You only need one. Okay, so you can just chop that off, the part that encodes for that protein, as an intron, and so now you can make this version of the protein. Okay, so good luck.